Hello, today we'll be talking about carbidopa and Parkinson's disease, unraveling mechanisms, and structural highlights. Parkinson's disease, otherwise known as PD, is a progressive neurodegenerative disorder that primarily affects movement control. It arises due to the gradual loss of dopamine-producing cells in the substantia nigra, or substantia nigra, I don't know how to pronounce that, a region of the brain responsible for motor coordination. The cardinal symptoms of PD include tremors, rigidity, bradykinesia, which is the slowness of movement, and postural instability. These symptoms result from the depletion of dopamine, a neurotransmitter critical for transmitting signals that regulate muscle movement. As dopamine levels decrease, the brain struggles to coordinate smooth and purposeful movements, leading to the characteristic features of PD. Carbidopa is a key therapeutic agent in the management of PD. It is often used in combination of levodopa, another medication that is converted into dopamine in the brain. Carbidopa serves a crucial role by inhibiting the peripheral conversion of levodopa into dopamine before it reaches the brain. This inhibition occurs in the bloodstream, preventing side effects and allowing more levodopa to reach the brain and alleviate PD symptoms more effectively. This combination therapy with carbidopa and levodopa represents a cornerstone in the treatment of Parkinson's disease, providing symptomatic relief and improving the quality of life for individuals living with PD. All right. Dopamine, a neurotransmitter, plays a fundamental role in motor control within the brain. It acts as a chemical messenger that transmits signals between nerve cells, facilitating communication in the brain's intricate circuitry. In the context of motor control, dopamine is particularly essential in regulating the initiation and smooth execution of movements. When we engage in physical activities, dopamine is released in specific regions of the brain, including the substantia nigra, to modulate the pathways responsible for coordinating movements. Dopamine helps fine-tune the balance between excitatory and inhibitory signals, ensuring precise and controlled muscle actions. In Parkinson's disease, a gradual loss of dopamine-producing neurons occurs in the substantia nigra. This deficiency disrupts the delicate balance in motor circuits, leading to the characteristic symptoms of PD. The decrease in dopamine levels results in a disruption of the normal signaling patterns, causing difficulties in initiating and coordinating movements. The dopamine deficiency is a central factor in the manifestation of motor symptoms such as tremors, rigidity, and slowness of movement observed in individuals with Parkinson's disease. Understanding the pivotal role of dopamine in motor control provides insight into the impact of its deficiency on the intricate neural pathways, highlighting why therapies that aim to restore dopamine levels, such as the use of carbidopa and levodopa, are crucial in managing Parkinson's disease symptoms. Carbidopa serves as a vital component in the treatment of Parkinson's disease by acting as a peripheral decarboxylase inhibitor. Its primary role is to prevent the premature conversion of levodopa into dopamine outside of the brain, specifically in the bloodstream. This is crucial because dopamine cannot easily cross the blood-brain barrier. Without carbidopa, much of the levodopa would be converted to dopamine in the peripheral tissues before reaching the brain, reducing its therapeutic effectiveness and causing undesirable side effects. The synergy between carbidopa and levodopa. Levodopa is a precursor to dopamine, and when administered alone, it faces challenges in reaching the brain in sufficient amounts due to the peripheral conversion. Carbidopa, however, mitigates this issue by inhibiting the decarboxylation of levodopa in peripheral tissues, allowing more levodopa to enter the brain. This combination therapy, often known as Cinemet, enhances the concentration of levodopa in the brain, where it can be converted to dopamine and alleviate the motor symptoms of Parkinson's disease more effectively. The strategic use of carbidopa alongside levodopa exemplifies a critical approach in maximizing the therapeutic benefits while minimizing unwanted peripheral side effects in the treatment of Parkinson's disease. Imagine levodopa as a key that unlocks the door to dopamine production in the brain, offering relief to Parkinson's disease symptoms. Now enter carbidopa, the crucial ally in optimizing this process. Carbidopa acts as a guardian, preventing levodopa from prematurely transforming into dopamine before it reaches the brain. This happens primarily in the bloodstream outside the brain's confines. Levodopa, akin to our key, is highly susceptible to conversation in the periphery, meaning it can be lost before reaching its destination in the brain. Carbidopa steps in as the protective shield, inhibiting the premature transformation of levodopa into dopamine outside the brain. This ensures that a more substantial amount of levodopa successfully crosses the blood-brain barrier, reaching its intended target in the brain's neurons. The benefits of this combination therapy are significant for individuals with Parkinson's disease by preventing peripheral conversion. Carbidopa enhances the efficacy of levodopa in the brain, allowing for more precise and controlled dopamine production where it's needed the most. This optimized delivery results in improved management of motor symptoms associated with Parkinson's disease, offering individuals greater relief and improved quality of life. The synergy between carbidopa and levodopa exemplifies a strategic approach in addressing the complexities of Parkinson's disease, providing a more effective and targeted treatment strategy for individuals living with this condition. X-ray crystallography stands as a remarkable technique in structural biology, allowing scientists to unveil the intricate three-dimensional architecture of molecules. First, a crystal of the molecule of interest is created. When X-rays pass through the crystal, they diffract, producing a pattern that can be analyzed to reconstruct the molecule's arrangement. The resulting electron density map unveils the precise positions of atoms within the molecule, providing a detailed blueprint of its structure at the atomic level. Chimera is an advanced molecular visualization program that plays a pivotal role in structural biology. It allows researchers to interactively explore and analyze complex molecular structures, offering a dynamic and insightful way to understand the relationships between different components of a biological system. With its user-friendly interface and powerful features, Chimera enables scientists to visualize, analyze, and manipulate the structures obtained through techniques like X-ray crystallography. In a live demonstration or display, we can use Chimera to showcase the bound structure of carbidopa, revealing how it interacts with its target at the molecular level. The visual exploration provides a deeper understanding of the drug's mechanism and how it precisely fits into its target, offering insights crucial for the development and optimization of therapeutic interventions. The synergy between X-ray crystallography and molecular visualization tools like Chimera exemplifies the transformative capabilities of structural biology, allowing us to unlock the secrets of molecular interactions and design more effective treatments for conditions like Parkinson's disease. In summary, carbidopa plays a pivotal role in the treatment of Parkinson's disease by enhancing the efficacy of levodopa, a precursor to dopamine, by acting as a peripheral decarboxylase inhibitor. Carbidopa prevents the premature conversion of levodopa into dopamine into the bloodstream, ensuring more levodopa reaches the brain where it can be converted to dopamine. This optimized delivery alleviates the motor symptoms of Parkinson's disease, offering individuals improved quality of life. Looking towards the future, ongoing research is in Parkinson's treatment is exploring innovative approaches. Scientists are investing in new drug formulations, delivering methods, and potential neuroprotective agents to slow the progression of this disease. Additionally, advancements in gene therapies and deep brain stimulation techniques are showing promise in providing targeted relief for Parkinson's symptoms. The evolving landscape of Parkinson's research underscores the commitment to finding novel and more effective treatments, bringing hope for enhanced therapeutic interventions and better outcomes for individuals affected by Parkinson's disease. These are my references that I used. Yes, thank you so much.